Hey, PBio, welcome to chapter 20, where we're talking about phylogeny and cladistics. So we're going to start off here with 20.1. And as we know, the tree of life is very wide, very old, and very diverse. And so we need to be able to classify them in order to understand which species are different, but also to understand the evolutionary history and map out their origin of each individual species. Luckily, a guy named Carolus Linnaeus, or good old Carl, came along in the 18th century from Sweden and developed the binomial system of naming things. So you've probably heard of a genus and species. So species means that they are capable of interbreeding and producing fertile offspring, and genus means they're pretty closely related and may potentially also be able to interbreed. So for example, with humans, genus Homo, species Sapiens. We are Homo sapiens. And this comes into a larger form of classification for life, going all the way up to domain, of which there are 3, down to kingdom, of which there are 6, phylum, class, order, family, genus, and species. And these are different classification categories. For example, humans are in the phylum of chordata, or those that have a spinal cord. Our class is Mammalia. And so you can start to see now all the mammals are grouped together. You can start to see how these groupings work to separate out these trees of life. Do you need to memorize this classification system? No. But I do strongly recommend you remember the phrase, dude, kindly please come over for good soup. Domain, kingdom, phylum, class, order, family, genus, species. If you can remember that sentence and you know roughly what those first letters correlate to, you should always be able to remember the order and know how close or disparate the grouping is, because obviously a family much closer related in evolutionary history than a phylum, since phylum is everything with a spinal cord. You and I and fish share the same phylum. So as we start to look at these phyla, we are starting to build phylogenetic tree, and we can start looking at these phylogenetics. And these are diagrams that show the evolutionary history using a branching pattern. And so what do these different branches really mean? Well, branches show the most recent common ancestor. So remember, in evolutionary history, there are species that existed in the past that no longer exist today. Some species from the past are still alive today. Sharks have been around for a long, long time. But this is kind of where two different species branched and separated away from each other on the tree of life. So again, this doesn't mean our most recent common ancestor is a chimp, but there was ape that was similar to chimps, similar to humans, and two groups evolved different ways, and one of those groupings became chimps, one became humans. So chimps didn't become us, we branched away from chimps. And so anywhere you see a branch point, that is the most recent common ancestor of anyone who comes after it. So this branch there is showing A and B, whereas that one deeper down, because off of that branch, A, B, C, D, and E all are there, it shows that it's an ancestor for all of them. And typically, the further back you go in your branching, the older it is. All right, 20.2, we're looking at just the genetic components, really. So these are phylogenetic trees that show genetic distances, how separately they're related. It also is typically going to show time since the most recent common ancestor. So a lot of phylogenetic trees will have a key, like you can see at the bottom, how many millions of years ago. And so you can see some of those branches happened more recently, some of those branches happened more distantly in the past. And so it tells us how long ago did that branching event occur where we now have two species where there was just one. And so this really helps us classify not only just based on species, but on time. And so what you might see in this class is a genetic table that's going to show either how many differences there are in the DNA, the genetic code, or maybe in the proteins. So remember, genes code for proteins. proteins are built by a building block called amino acids. So how many amino acids in that protein are different? So more differences means they're more distantly related. So you can see in this table that E. africanus has only one amino acid difference from E. ferris. So on our tree, they're very closely related. Whereas E. ferris and D. polyepis have 21 amino acid differences. So they're going to be a lot more distantly related. So you can see E. ferris is on the opposite end as D. polylepis. So more differences, more distantly related, we can start to build our trees. All right, 20.3. This is going to be cladograms. So cladograms is a system kind of like with our phylogenetic tree where we're placing species into groups called clades that all will share a common ancestor. So a clade, they all share a common ancestor there. Cladograms and phylogenetic trees, for this class, for the most part, you can use them interchangeably. They are different where cladograms are going to be more focused on clades and phylogenetic trees are going to be a little bit different, but don't worry about those differences today. They're going to be semantics for us. So one common way that cladograms will be used is not looking at genetics, and this is especially common for things before we really were able to sequence genetics, was looking at characters or traits that all species after that point on the cladogram would share. So at these branching points, after a branch, there's going to be some new character, some derived character that everyone after it in the clade is going to share. So for example, this clade of mammals that you can see with the primate and the rabbit, hair was the shared derived character that developed. And so only those species are going to have hair. And so you can see on here that these characters are how we're separating out these organisms. You'll see shared ancestral character. These are a character that originated in an ancestor outside the clade. So if our clade or grouping is just mammals, vertebrae go way, way back. Lots of different species have vertebrae. So that would be a shared ancestral character, not unique to our clade, but it's present. A shared derived character is just unique to that clade, so like I talked about with hair. So another way to build a table here is looking at whether or not they possess a certain character. So these character tables you may see. You may see a check and an X for has or doesn't, or a plus and a minus for has or doesn't, or here you see 1 and 0. So 1 means it has it, 0 means it doesn't. And you can see that exact same thing there, where as those traits developed, you can see those characters placed on those branching points. Everything after that branching point has that trait, like jaws. Everyone but the lamprey has it, so everyone after it on the tree would have that trait. All right. When we're looking at these clades, there's different ways to group them. And so we're going to be talking about 3 groupings: monophyletic, paraphyletic, and polyphyletic. And we'll break it down. Monophyletic means everything in the group has a common ancestor. All descendants of that common ancestor are included in the group. So this is really used to discuss those shared derived characters that we talked about. So for example, the amniotic group here all have an amniotic egg. So frog and fish don't have those. So our turtles, our mammals, our mouse, humans, all of these have an amniotic egg. Birds would be in there, reptiles. A monophyletic group, if we were to simplify it to just like your family, maybe it would be like you and your siblings and your mom, but we're not counting your aunt and your cousins because that's a different branch of the tree. Maybe they have some different traits. But we can see here that they all have the same ancestor, and everyone from that most recent common ancestor is included. Now we're going to differ it a little bit with paraphyletic. So this is very similar to monophyletic, but now we don't include everyone. So they all share a common ancestor, but we're not including everyone. So this is used when we want to talk about maybe there's a group that has diverged from the group. So that amniota, again, we can see that amniota group. If we're just talking about the reptiles, so that's going to include those crocodiles, those turtles, those lizards and snakes. But birds separated from that reptilia pretty recently from that crocodile family. And so because of that, if we're just talking reptiles, we can't make a monophyletic group from their most recent common ancestor because that would include birds. So we would talk about a paraphyletic. So this is when somebody in that clade, in that monophyletic group, has diverged so much, we don't want to talk about them, so we're going to exclude them. Polyphyletic is going to be when we're talking about totally different lineages, or we have to go way far back and to find a common ancestor, and there's going to be a lot of exclusions. So this isn't paraphyletic because we're not like excluding one or two, we're excluding a lot here. So this is going to be used a lot with convergent evolution. So for example, if we wanted to talk about from this amniotic group, organisms with wings, we might be looking at those birds and like some mammals like bats that we're talking about and grouping together. So you might be looking at convergent evolution here with those analogous structures, for example, where you're building a polyphyletic group. This is used when you've got some groupings that are way different than everybody else in that clade. When you're building your trees, you want to make sure you adhere to the principle of maximum parsimony. And what this means is we basically consider a trait probably only evolved once. It doesn't appear, disappear, reappear, disappear, reappear, disappear. We're going to assume it only happened once. Maybe it appeared and went away, like with whales, those vestigial structures like legs. But it's not common that it's going to like appear, disappear, reappear. So maximum parsimony shows the fewest mutations needed to make a tree. We want to have the fewest derived characters, the fewest mutations to build our tree. We want to go as simple as possible. So that's going to be the principle of maximum parsimony. Always try and build your tree as simple as possible. Don't make it so there's multiple changes. That's basically it for phylogenetics. Make sure you participate in our activities in class. That'll hopefully clear it up with Candy, and we'll see you next time.